Uh,
Hello, everyone. Welcome to the MIF++ seminar. It's a pleasure to introduce Dr. Ezra Peisach from the Rutgers University and Worldwide Protein Data Bank, who will talk today about the PDB validation process. So, Ezra, if you feel that your slides are ready, yeah, feel free to start. Over to you.
Good afternoon, morning, wherever you are. My name is Ezra Peisach. I'm a biocurator / software developer / dictionary maintainer at RCSB PDB. Just to set us up in the get-go, I don't mind if you interrupt for questions. I could make my slides go faster, slower, talk faster, talk slower. I don't care. So whatever works for you. So to set up the stage a little bit, I'm just going to say that the Protein Data Bank was established in 1971 with 7 original structures, and it's a single global archive of macromolecular structural data. Structural data. We were the FAIR before FAIR was common. Data sets were distributed way back when on 9-track tape, later on DVDs and FTP, and now the variety of mechanism to get them. We have over 200,000 structures in the archive right now. We are collaboration. So while I represent RCSPDB, we are the United States contingent, but we have collaborators in the UK at PDBE, Japan PDBJ, an associate member of PDBC, and we have 2 smaller adjacent archives, EMDB for electron microscopy data, and BMRB, which handles NMR data as well. But the coordinate section and models belong to the PDB. So I'm going to go over, this is a general high-level overview. So I'm going to talk about why is validation by PDB necessary. I'll give you a little introduction to structure determination. It's not going to be exhaustive, but it'll give you an idea of why there are issues and errors that are sometimes introduced, not intentionally, by depositors while they're working on their structures, because it's not a trivial task to do. And then I'll discuss the validation pipeline that we are using at wwPDB, and you can see how we do things. So why is validation necessary? Macromolecular structure determination, as I said, is difficult. For small molecule crystallography, you have enough data that you could actually determine the positions of every atom with high precision. With macromolecular structures, you are underdetermined in terms of the amount of data you usually have. So for X-ray or EM structure determination, I call it a guided rebuilding followed by automation of refinement. And errors are introduced. But I would like to just point out that the PDB, we are not the structure police. We are an archive. We have to take everyone's data, but we'd like to help the depositor to get it right. If we can tell them, hey, we know something that looks wrong here, a lot of depositors would rather have that information told by us versus someone else reading the paper, looks at 2 years later and say, you got the chirality of that ligand wrong. They would rather have us tell them that, and then they can make corrections beforehand. And that's sort of the goal here, is that we providing feedback early, depositors correct structures before the public sees them. We also provide a role in terms of, we give them a validation report that could then later be shared with journal reviewers. I'll get more into that later. So I found a paper from 1996 talking about a program by the Sanders Group called WhatCheck, and they analyzed the 3,400 structures that were in the PDB at that time. You can see how we've grown in a number of years, and they found a number of issues. There was inconsistent symmetry space groups. There were DNA amino acids where there should not be. There were clashes of atoms near symmetry-related axes. They had occupancies that added up to more than 1. They found large bond length deviations from what they call large bond deviations, more than 4 sigma from reasonable data sets, large angle deviations, and they found atoms which had that were clashing with each other. And to put things in perspective, the field of science and structure determination has changed in 50 years. A lot of these would probably have been detected now, or even the refinement packages would not have allowed these to happen. But it is where we are. So they came up with a package to go through and make it easy for an end user to take a structure, hopefully the person working on their structure, and run it through their software and identify issues before it got out to the public.
Ezra, could I
Yes.
ask here a short question?
Yes.
This particular check it was done for this number 3,442 structures.
Yep. That was I believe, I did not go back and check, but I believe that was what was available in the public archive in 1996.
So this was the full PDB at that time.
I believe so.
I see this number of structures.
I'd have to double check. I haven't looked at the numbers, but they suggested that.
And approximately, say, what was the percentage of structures that required that had the issues listed?
Their paper over here, which is mentioned here, talks about the actual numbers. I didn't look at what percent. They didn't refer to percentages. They said, there were 15 cases. I can't remember. It was 15 cases of inconsistent symmetry, 10 D-amino acids. I don't know, but were any of them overlapping with each other? I don't know.
Okay.
Because they didn't have it was a 1-page or 2-page paper in, I can't remember, in Nature. So they didn't go into very much in the details of here's the list of entries. They didn't call out anyone and say you made a mistake. But these are the data that were deposited, some of them say 30 years ago, and the PDB is not going to make changes to their data. So we hopefully, and you can see by providing validation, I'll show you a plot later on, people are willing to try and do better, and they are doing better now.
Right. So these found errors were corrected later?
No, probably not.
No.
The mechanism that the PDB has, or at the time, was that the depositor of record, principal investigator, has the option of submitting new coordinates, a new model, to replace the existing one. And we could obsolete an entry and say that it's replaced by something newer. And so if you say put in your publication, you have structure 1ABC, we could say 1ABC is now replaced by 3ABC. And if you try referring to the old structure on our website or our partners' websites, you'll automatically be redirected to the new one. But the old data are still available. And that was an important consideration that, it may reflect what the publication really had, but they wanted to correct it. We now have the mechanism that if the depositor of record would like to resubmit new coordinates without changing any of their experimental data, but keep the same accession code, they can do so, but we keep a versioned FTP server, versioned archive, where the older versions are still available, just so that you could see if you want to do any analysis. The other thing that we provide is an annual snapshot, that if you're doing a data analysis, and if you say, use the snapshot data, which is produced the first week of January every year, that data set will be consistent. So if you use that in your publication, 5 years later, you can go back and say, here's what the state was at that point.
Yes, of course, it's very reasonable to keep old versions, but could I clarify? So the new versions for all these issues were submitted later? No.
They could, but whether the original depositor did so, I'm guessing most of them did not. So we have two types of depositors. We have depositors who really care about their data. If they saw that there was an error, they would rush out to fix it. There are others where the structure was only a part of the big overall experiment. And if you have, we have a clash between a lysine on the surface that's nowhere near the active site, how important is that to fix to them? Because it's not going to change their story. From their story, the ligand bound in the active site, it did how they function the way it did. It may not be the best solved structure, but I just we can't control what they do on that front.
Okay, so when these errors are reported, for example, in this Nature paper, so apart from this Nature paper, is this information recorded anywhere in the PDB?
No, because we do, yes. So we'll get to that, but in terms of what the validation reports do report a lot of this information, so it is available publicly. It is not associated necessarily with the model file. We do have the ability to put caveats in the structure if we notice there's a problem. So the most common chiral issue that we put in as a caveat record would be if there's a chirality issue. And this happens frequently with sugars, where they detect, if we detect a chirality issue relative to ligand, so NAGs, for instance, C1 frequently is the wrong chirality. Well, not anymore, but it used to be, where I worked with a depositor. We had to go back and forth 9 times to get the chirality correct. And they were trying to do it, and they were working on it, but the software they had was not doing a good job at identifying the problems that we could identify. So then they give us a new model coordinates. We look at it and say, okay, here are your errors now. Send it back to them, and they work on it and send it back to us. So those who care will try to get that, but there is no one universal place that says, here are the issues that we've identified.
Okay.
Okay.
Thank you, Ezra.
Moving along. So as I said, why is the structure determination difficult? As I said, small molecule crystallography are usually at very high resolution, better than 1 angstrom resolution, and you have sufficient data to find all coordinate positions, temperature factors, anisotropic factors, I have a typo there, the position and occupancy. Macromoleculars were underdetermined. If you're working at 2.5 angstrom, 3 angstrom data, you're not going to have all the data that you need in terms of to uniquely solve your structure. So we introduce into the refinement process physical information such as bonds, dihedral angles, improper angles, etc., where we know what they should look like based on chemistry or small molecule crystallography. We know what a peptide bond looks like. We introduce that into the structure solve. Well, not we, but people developing refinement software introduce that information into the refinement process. Some refinement packages would also introduce secondary structural elements. That was an option in some packages. If you know there's an alpha helix, you know what the hydrogen bonding pattern is going to look like, and you could add that in as other restraint information for solving. So I'm just going to give a quick few-minute guide to structure determination, just so you can see how it's done, and you realize where the balance goes into solving a structure. So this is a very simplistic cartoon. You get a crystal, you have a diffraction pattern, you've indexed all these reflections that you see in your diffraction pattern, you get phases. I'm not going to talk about how you do that. You generate electron density maps. You fit your model to your electron density maps, and then you do some form of refinement that tries to do a little better positioning of and fixing up your geometry. And you repeat till you're done. So just to show an example, here's an electron density map from showing you a serine residue, and you can see what, I won't go into very much details, but blue is sort of representing what you have and what corrections you might want to make to your model. Green is density that you've under accounted for. Red is electron density maps, which shows where you may have over counted. And this would suggest that the serine side chain would want to flip over to where this green density is. And this is the type of iterative process that as your phases improve, as your model gets better, you improve your phases, you get better maps, and then you could make corrections of this nature. We're not going to talk about, it is also possible to bias your data. If you could put in waters into random places, you could wind up locking yourself into a structure. So this just show you this is a theoretical computation of what electron density would look like at different resolutions, just to give you a sense of, and this is perfect data, and you can see that there's a bit of slop. You know, 6 angstrom, you could do anything pretty much in terms of you have no idea what your backbone is going to look like. As you move towards higher resolution, you can refine generally where the atom positions are, to when you get to extreme high resolution, you'd be able to definitely say you know where the atoms are. So somewhere in a lot of structure determination is somewhere in the middle, and you're trying to say which way does this go. What you're not seeing is connectivity issues. Say if you have a long side chain amino acid like lysine or arginine, is not atypical for the middle of the electron density in the middle of the side chain to disappear, because your side chains are mobile. And if you don't have it in a crystal structure, is an average of states, and if you don't have enough occupancy, you may not be able to resolve it. So to improve the position after you've done a manual fitting, there is a refinement process which tries to correct your physical geometry issues and fit to electron density map. And most of the current refinement methodologies use an energy minimization scheme in which you are creating some large total of how much energy would, and I'll go into details of bonds, angles, dihedral angles, and proper angles in X-ray terms. And then you're balancing a physical geometry and your experimental data. This energy term is usually a very simple approach, Hooke's law, which we all learned in physics. I learned it in high school, which has to do with the deviation of a spring. A spring has a natural location it wants to be at. If you pull the spring out, it wants to pull your atom back. So this is where we're talking about ideal geometry for what various bonds would look like. And this is all weighted. You can throw in a Leonard-Jones 612 potential to represent when atoms get too close or further apart, and try to model that as a collision, prevent that from happening. And you can
a quick question about the slide with Hooke's law.
Yes.
The x0, x0 value. So this is an ideal distance, I guess,
right.
between atoms, right?
Exactly. So that's your resting state.
Mm-hmm.
So if you're so consider a carbon bond here. Say you have an ideal length, and you say what I did some manual moving of this atom and I pulled it out. You'd want to pull it back into more of an ideal position. But what this allows for is you're not locking yourself into this ideal value. You're allowing, I can have a little deviation, and that's still okay. Mm-hmm. There's a penalty for it because there's a little bit more energy to it. But what's a tenth of an angstrom? Well, at a 1 angstrom structure, a lot. If it's a 3 angstrom structure, there's a lot of room to position things in there.
I see. Ezra, these ideal values, are they known?
The ideal values can come from a variety of places. I would say for things like standard amino acids and things that have been observed a large number of times, these values have been put together from data sets, usually from small molecule crystallography at high resolution. Mm-hmm. But for other experimental ones, I was working on a project once where we had to find what does a carbon-fluorine bond look like in this trifluorine look like? What are the bond angles going to look like? And that people used to do real literature searches or looking at the Aldrich catalogs to look at spectra and try to figure out what are the ideal parameters that you want to use for this if you have an interesting ligand. So nowadays there are more automated methods that use quantum mechanics and try to come up with a better a model for that. But 20, 30 years ago, that wasn't an option.
Okay, thank you.
So it comes from different places. Another approach that you could use nowadays is to search the Cambridge Small Molecule Database and see if there are pieces that look similar to what's been observed. And we actually use that, and I'll talk about that later. But say you have a small fragment of a ligand and it looks similar to something else that's been seen and crystallized in the small molecule database, that might be good parameters for you to work with. So that's been something that's physically observed in the crystalline state. But a lot of these refinement packages have standard set of parameters that are all balanced properly to be consistent. So then we have the X-ray term. And I'm not going to go into detail, but we have our observed data. We can calculate what our data should look like based on, there's a way of calculating what our data should look like based on our model. And it's another energy term. We're trying to minimize the difference between them. It'll never happen, but that's part of it. And then we're putting it all into one large energy term, and then it's a minimization function. And these are weighted. So you have a trade-off between, do you want to go stronger weight towards the X-ray data? Do you want to go more towards ideal geometry? And there's a balancing game between the two. But what I'm trying to show is that you might be able to have a deviation in a bond length in order to satisfy the X-ray a little bit. And you're trying to just get an overall balance that looks good. So wrong chirality could go in from an energetic term. There is a penalty for that, but usually these methodologies will not flip the chirality of an atom. So if you manually move it to the wrong place, it will likely not flip it back, because the iterative refinement process to move it stepwise to get back to ideal. Now there will be even worse energetic terms while it's going through the transition. So you've got to be sort of in the neighborhood. And as I said, refinement is iterative. You adjust your model, you refine, calculate new phases, new maps, and repeat. And it's dangerous that you can overbias yourself. So the question is, when are you done? People often use an R factor to say my data my refinement process has converged in a final place. And so people say if you get less than 20%, oh, you're done. You can over refine your data. So a methodology was developed by Axel Brunger around early 90s was to take some of your reflection data set and put it aside and don't refine against it. You can generate maps against it, but you don't refine against it. And so that's an independent test set. And you get a similar type of calculation. You expect that to continue going down. If you start over refining, you'll see the R-free starts going up. But hopefully you're done when it agrees with known biophysical data. So does it make sense? Are there right-handed helices? There was an early publication of X-ray crystallography where in the paper there were left-handed helices. I mean, there were right-handed helices instead of the standard left-handed helice. And a reviewer who had known more about structural data would have said that's wrong. And so it was clear afterwards. Does the oligomerization state make sense? You expect to be a tetramer in solution. If you can't see a way of building a tetramer, there's probably something wrong. If there's metal binding, you probably have some biophysical data say we know who the metal binding partners are. Are they interacting with the metal? Do they look correct? If you know where there's an active site, you know where the critical residues for activity are. Do they make sense? Are they near each other? If the fold is similar to a similar structure in a similar class, so for instance, you have a serine protease, you know what serine proteases generally look like. Do they look similar? Yes or no? And you have good geometry and statistics, which is what we'll talk about for the rest of this, which is Ramachandran angles, clashes, good rotamers, etc. So now I'm going to switch back to the wwPDB validation process. As I said, we do not reject structures based on data quality. We would like the reviewers of an entry to make that determination for us. There have been a number of structures where, well, not many, but people have questioned. They said there was a particular ligand and there was no electron density map to support it. Well, it'd be nice if the journal reviewer had the view that we had of electron density maps to make that determination for themselves. And that's something we put into the validation report. I'll show you that later. So we started producing wwPDB validation reports. Were produced with the one-step deposition and annotation system in 2014. So just as a reference point, in 2021, 2 years ago, we had around 14,000 structures deposited, and 20% of them had coordinates replaced after deposition and after we provided a validation report. We hope that a lot of those corrections were because we gave them feedback and they said, oh, I could do better with this and fix it. We don't know for certain what the reasoning was. We don't ask for the rationale. They say, I like to give you new coordinates. We don't ask what the reasoning is. As I said, we also encourage journals to require a validation report from us. So if you're reading a journal, a paper, nowadays structural data is just one component of the publication. You might be using 3 or 4 methods, and the journal has 3 reviewers, and maybe none of them are structure-based people or have a background in structure determination. So we'd like to produce a report that they could read and hopefully be able to do some form of evaluation of whether the structure was done properly. We have agreed with many of the journals, or the well-known journals, now require validation reports that produced by us. We are never going to provide data or validation reports directly to the journal. That is up to the depositor, but we are giving them something that they could sort of submit along with their paper. It's just sort of additional information that doesn't get published, but it's just for the reviewers to look at. And the goal is that for end users, it's something that they could look at and say, if I have 2 structures, which one is done better, without going into nitty-gritty detail. So most of the users of at least the RCSB website are non-structural biology experts. So we have to produce something that they could look at and help to interpret the data and whether the structure was done well. So this just show the concept of if you had an electron density map, it's an interpretation. The PDB entry model file that we provide in our archive is a interpretation of the data. You can interpret it multiple ways, and hopefully the interpretation is good. So just to give a bit of other background, what did validation look like at the PDB? So through the 20th century, so 1990s, at least the 1990s, I submitted my first structure, and I got back, here's a limited list of validation issues of geometry. I said, I have a few close contacts. Our waters might be on a special position. And that was about it. Everything was left up to the depositor. In the early 21st century, the validation reports we provided were better. We gave pictures of Ramakrishnan plots, which showed where are you in a favored region, crystal packing analysis. And later on, when structure factors were required in 2008, we did a structure factor versus data analysis to see could it agree with it. I'm not saying that before 2008 people were not submitting structure fact files. It was then required. So there are structure factors, the experimental data going back to the 70s, but not everyone submitted it. I always took the approach of it's the ultimate backup location. If a hard disk fails, they have a copy of it there. So it's out there in the public. So when trying to come together what to do with validation, while we think we're experts, we're really not the experts in the field. We brought together various task forces of people who really knew the various experimental methodologies and had them tell us what we should be using for validation in a standardized way that could be used worldwide. So in 2008, we had an X-ray validation task force chaired by Randy Reed. 2010, there was a map and model ones by Richard Henderson and Andre Sali. It was at the same time, but we divide up into maps versus model group. And in 2013, an NMR one. And these recommendations were mostly incorporated into the validation task force, into the validation reports, which I'm going to talk about in a minute. Our www.pdb.org website, I'm just showing here a picture of it. It shows where you can get more information about the various working groups, including model CIF, hybrid integrative methods, and their reports and publications that came out of these various task forces that were put together with us helping sponsor. So as I said, the wwPDB validation report was introduced in 2014. Every structure in the public archive has a validation report right now. It is a watermarked PDF validation report. We give several flavors. The public sees only one, which is the final one, but we provide one to the depositor prior to their submission, so that they can hopefully improve things before we even look at it at all. We give it to them after annotation. This is what we want for journal review. That's after we've processed any ligands. We do all the processing that we would do before it goes out to the public. This is what we want them to submit to a journal for the reviewers to look at. And then we have the one that goes into the public archive. With each validation report is an XML data file, which includes the data that are present in the report. Pretty much every number that's that you'll see in the PDF validation report is available in XML data file for data mining. Certain molecular reviewers, Malstar, for instance, available from we use for graphical representation of our website, will use this data to indicate outliers or potential issues with the model. Coot, I believe, which is a part of the CC4 package for their graphical rebuilding, I believe could parse this XML data file and provide and display the results as well. So you could then you're looking at structure, you could look at what has been identified in these files. The wwPDB in the last few years has also come up with, instead of XML format, an MMCIF-based validation data file, which contains all the data will continue to move. Until we recalculate every entry in the archive, it will not be present there. So I would say every entry that's been touched in the last 2 years has this data available. But by the fall of this year, these such files will be available for the entire archive, because there's going to be a recalculation of everything. So what are we checking? Well, we look at the fitting quality. How well does the data fit with the experimental data? We look at global and local data. geometry analysis, how well is it matched to known geometry? We talked about standard amino acids, but how well does it fit to other ligands that look similar? And then we have method-dependent analysis, which would be other features that are used by the particular methodology. This is sort of a high-level pictorial representation of what we're validating. So the basic flow is that we have different steps. We look at the polymer geometry. We then look at the overall data quality, which is independent of the model. We will look at the model fit data, and then we'll look at the geometry, the ligand geometry, and then we'll give you a comparison to all the other entries. So for geometry analysis, the primary use is mold probity, developed by the Richardson lab at Duke University. It's now available in CCP4 and Phoenix, as well as independently, and that is used to examine clashes between residues. The methodology tries, I'll talk a little bit in a moment, but tries to put hydrogens atoms in position and then looks to see where side chains might clash. For data quality, we're using XTriage from the Phoenix software suite. Visual analysis is something that was developed by EMDB, which looks at electron microscopy maps. We perform chemical shift analysis with a variety of programs. For fit of model to data, it's EDS, other CCP4 programs. We look at volume inclusion, and now we're moving towards analyzing the restraints that are provided with NMR structures. And then ligand geometry, we use the Cambridge Small Molecule Database program Mogul. And I'll highlight what you could see in each one of those. All this information is summarized at the top of the validation report in what we call a slider. And this was developed, the concept of a slider was developed a number of years ago, where you're looking on this one slider, you could see, I'll go into what each one of these means, but the R-free score comparing what is the percentile that I have? I have a value of, say, 0.209. How does that compare to all X-ray structures in the archive? And how does that compare to structures of a similar resolution? So a higher resolution structure, you expect to have certain qualities that are better, but it's going to look very different than the quality of how well your model fits the data at 3 angstroms. So it's only fair to compare you to your local neighbors, so to speak, in that sense. So let's look at what some of these fields are. So as I said, how well we fit to the experimental data. RSRZ is how well we fit to so the R-free is how well does the model fit the experimental reflection data. Real space R factor Z-score is how residues fit within the electron density. We look at atomic clashes. We look at the main chain torsion angles for Ramachandran analysis, and we look at side chain angles. So the R-free, that's just a number. I showed you the value for what an R value was, and the R-free serves to keeping it aside. And that's sort of an overall fit of how well does your model fit to the data. So that's a single value. So what we have here are 2 different examples. One is 7BYQ. And if you were to look at the electron density map, you'd say it looks very ropey. It doesn't really seem to be convincing that it fits the model well in this region up here. You can see other regions in the lower left, which looks better. In the upper right is 2HYV. And you can see how, for most of the model, you can see it fits very well in electron density, but you see even a surface, you can see a surface lysine. There's no electron density, which I mentioned that happens, and it looks like but overall it looks like a better fit. The RSRZ outliers look at a per residue level of how well does a side chain fit into the electron density. And you can see here, the tryptophan does not fit as well as say, looks like an arginine fits it in this place over here. So better and worse. For geometry, we are looking at clashes. I mentioned that we are looking at this from mold probity. So you could see here a clash between residue, because you could say, hey, it looks like they'd be butting up against each other once you start putting hydrogens on there. We look at sidechain, which is a torsion analysis based on favored positions. There are databases which list what are the favored sidechain conformations and how well do you fit against that. Doesn't mean it's wrong, it just means it may be unusual, and you may want to look at it. Frequently, when I was solving structures, I'd get a report by that, and I'd go back and look at it, yeah, there's a better rotamer that could possibly fit. And I said, Ramachandran, we're looking at the phi psi angles, cloud area. For small molecules, we look at how well does it fit, a ligand fit into the electron density. And this is something which a reviewer might want to look at. So you can see here, I don't know what the 4WI is, but this is some inhibitor and fits very well into electron density versus this other one, which DIF, which one would argue fits poorly. And there's some scores which RSR, RSC, real space R factor, and real space correlation coefficient. And you can see the higher the number for the correlation is better fit. We also look at the geometry of small molecules. So this is using the Cambridge crystallographic data, using a program called Mogul, which tries to say, let's break our structure down into fragments, our ligand down to fragments. Has that been seen, that fragment, within the small molecule database? And then it tries saying, how do we look in that stage? So we generate a distribution of 1,000 entries that's randomly chosen by their software. It will look like a plot like this, and you could say, oh, looks like the ideal bond length would be around 1.4. Our value is 0.9. That's probably not right. There may be things that we don't know about in terms of charge and everything like that, but it gives you something that you may want to look at. So our validation report will also show what it considers are outliers in a 2D flattened representation. So it's saying this bond there is unusual in terms of what's present in there. And some bond angles look unusual compared to other small molecule fragments. And so hopefully the depositor will come back and look at this and say, I either did a good job and that's the best I can do, or maybe there's something I could look at, and maybe my parameterization of my ligand may not be the best. And just want to point out, the validation report is more than just pictures. We also are providing the real numbers. So we could say, like, molecule 1, chain A, there were 18 clashes. We added 1,300 hydrogens to the model. There were 1,349 without hydrogens. And then there were 18 clashes. And then we put them in how bad the clashes were in order. So you could look back and say, oh, these two valines, they might be a little close. The citate of valine 40, it might be a little close to the backbone valine. I want to look at that and maybe make a change, or you may decide, at my resolution, I've done the best I can. We are also providing electron density images for ligands of interest. So we're not going to do it for every ligand. We wouldn't want to produce them for magnesiums, but if you say in the deposition process that this ligand is important, we'll produce an electron density plot, and you could then, that goes on to a reviewer who can then say it fits well. So this is a way of a reviewer being able to say, did the depositor do a good job of solving their structure? Electron microscopy has their own plots displaying quality, but this is an example showing the fit of a model to the electron microscopy map. And this one, it looks pretty good. Sometimes you see domains that are outside, and sometimes, but this allows a reviewer or an outside user to look at this file and say, hey, this looks like it might be a good map fit. So then, how has this helped improve structures? So this was an analysis done by Cheng Shah in our group. He took a look at what he perceived as the impact of the validation report on X-ray structures. And what he has plotted here is clash score and sidechain rotamer outliers for what he was calling legacy entries, which were data deposited between 2012 and 2013 before we provide these validation reports, and compared to the one-depth validation report, which was from 2014 to 2015. And what you could see, graphically, you can see, oh, side chain rotamers, we're getting more that have fewer rotamer outliers, and we have fewer clash scores, and that shows up where the overall archive percent of he came up with the quality score. The medium one was lower than the 2012, 2013, and then 2014, 2015 was improving. So by providing this feedback, we show that the depositors are able to improve the quality of their models before it goes out to the public. Every partner website, RCSB, PDBE, PDBJ, all provide the slider information and the ability to download the full report that I was alluding to from their website. RCSB provides a button, one-click button to display the 3D report graphically and indicate the outliers in real time on the 3D display. So say you have some data that you want to validate, can you do it? And the answer is yes. So we provide three processes. For most users, they could use our anonymous standalone user interface based validation one, where you could go through a graphical interface where you upload files, experimental data optional if you just want to do a geometry analysis, and it'll come back with a validation report. We also provide an anonymous API based service if you want to do programmatically. We had someone who generated validation reports on, I can't remember, it was 110,000 structures, which were some form of novel refinement method he was working on. And he let me know he was going to be doing it before he started up, and said, go for it. And then there's the actual validation report during submission process. And this is, again, the wwPDB.org website, in case you want to go find where this information is. Again. So, in wrapping up what I've been talking about, solving structures can lead to real errors, not intentional. We assume that most of the people who submit a structure, they're not trying to commit fraud. It's just honest mistakes. And can we help them improve their deposition before it goes out to the public? We show that the validation reports how well the structured coordinates fit to experimental data and how well it fits to well-known references. As I said, we provide to the depositor at the time of deposition, and then the depositors provide to the journal when they actually submit their paper. Even after they submit it to the journal, they could still do revisions to their models and structures up until the point of release. And people come back and they sometimes say, the journal felt that my high resolution cutoff was wrong. I should cut it back a little bit lower. Can I do it? And the answer is, of course. That's the way the process is supposed to work. And the results are, as I said, presented on the wwPDB partner sites. And the goal there is to help a user coming in to understand the quality of a structure and in a non-judgmental way. So I'm just going to say thank you for your time. I'm just going to point out this is the team that works at RCSB PDB. Our collaborators at the wwPDB have their own teams of similar sizes. Our PI is Stephen Burley, and Emeritus PI was Helen Berman. Andre Sali is a collaborator from the UCSF site. And this is our team as it is today. And with that, I'll ask if anyone has any questions.
Yes, Ezra, thank you very much. So first, let us thank Ezra for his nice presentation. Ezra, if you could keep your slides, it would help us to ask questions. But let me
press the magic button, it goes away. Okay.
Yeah, let me stop the recording so we could have an informal discussion.